Hello everyone. Today I will discuss about bacterial growth curve. This is very much important topic in microbiology. To describe the growth and the bacterial uh, development of their cells, we have to know the growth curve, and we have to know this growth curve for the efficiency of any antimicrobial agents that will destroy the bacteria and to help to cure the animal that has been affected by various diseases like bactericidal diseases. So let's move on to this very much interesting topic. At first, we are looking that there are a growth curve here. This is generally a growth curve. To focus on this growth curve, we have to understand the pattern of this. Look at this pattern here. Here, this is the very much lower pattern, and it is growing above. And this is the stationary. In this phase, this growth curve is not increasing or decreasing. This is state here, and after that, after some moments, it is also in decreasing in a slightly pattern move. So, in this curve, we have categorize the four phases to describe this growth curve easily. Number one is lag phase, number two, log phase, three, stationary phase, and the final one is death or decline phase. So we have to learn what are they indicating and what is the condition of them. So number one, lag phase. In this phase, we have to, before growing a bacteria, we have to culture in a culture media. So after culturing in a media, the bacteria will take nutrients from that culture media and it will uh, start their development. But in this phase, there will no number of increasing cell. There will be only the size increasing. At this phase, there will no multiplicative site. And if there is no multiplication, then we can say that there is no binary fission, as bacteria will grow from the binary fission. And if there is no binary fission, so there will no increasing uh, in the number of the bacteria. Okay. Then the second one is the log phase. The log phase. The log phase is their exponential phase. Okay. Look at this phase. This is from this state, it is exponentially increasing. We can say that it is logarithmic phase. It is logarithmic phase. And in this phase, we can see easily that it is increasing. So in this stage, it is increasing actually the number of the cells. I mean, there is binary fission. So in this stage, after lag phase, the binary fission is started, and then the number of cells is increasing. And here, as there is binary fission, so the size will be decreased because as they took nutrients in the previous phase, as there are no binary fission, so there were no chance to lake of their nutrients. But in this phase, they have to uh, compensate at this, and so they become increasing in number and there will be no increasing in size. Okay, number three is the stationary phase. Why is the stationary phase? This is very much interesting question. There is, if there is very much binary fission, we can say, we can write here as there is very much binary fission here. So if there is very much binary fission, there will be toxic, very much toxin or toxicity very much. So if there is toxicity very much and the nutrients will be lower here, nutrients lower. So nutrients lower and also the mm, place, place or area become also lower. So if there is much lower, so bacteria will not grow very much in this state as there are no nutrients and no place required for the essential growth of them. So in this stage, if their nutrients become lower, so they started to live in the stationary phase. And if as they are present in toxic or toxins, so they in that stage that before because of the presence of toxin, the bacteria will die. But most of the bacteria will not die in this phase, and some other bacteria will be definitely the log phase. From the log phase, there will be binary fission, and in this phase, there will be toxins. So the size, the number will be the equal stage as the living condition. Living condition is to diet condition is equal. So as this is equal state, so the curve remain in static position. This is the stationary phase. Okay, and the number four is decline or death phase. this is very much recognizing that it is lower. As it was exponentially higher, this is lower because of the I have described in the previous phase. This is toxins, nutrients, and required place. The toxins is increasing very much. The required place is decreasing, and nutrients are also decreasing. So there will be no question for any binary fission. The phase, the size will be also in lower condition, and most of the bacteria will die. If any bacteria is not died in this death or decline phase, so we can say that this bacteria is antibiotic resistance. And if any bacteria is not died in this phase, so we cannot apply any antibiotic that was applied before to kill the bacteria. So this is very much recognizing for the antimicrobial test. And in the stationary phase, we can apply the antibiotic stage, because in this stage the bacteria are very much active to binary fission, and they are taking very much nutrients to multiply. So in this stage, if we apply the log phase or stationary phase, actually log phase is very much recognizing to uh, apply antimicrobial agents to, because they are taking very much nutrients here. So if we apply any antibiotics in this stage, then the bacteria will be died and the patients will cure. But in death phase, if there are any bacteria that will survive from this phase, then we cannot apply any antibiotics. So this is the overall phase of this. From the next slide, we can see there is literature of this that I have described, that in maturing and not able to divide, that's binary fission is absent. And this exponential phase, there is binary fission is applied there. Okay, and it's number three, stationary phase. The growth rate shows as a result of nutrient depletion and accumulation of toxin products. The phase is reached that the bacteria begin to exhaust the resources that are available to them. And at last, the dead phase, there is run out of nutrients and space, and the bacteria cells will be decreased. So this is the overall description of bacterial growth curve of phases that will help us to describe the antimicrobial agents and the bacterial growth and development. So that's all for today. Thank you very much.